Hello, I am Anthony Vandermeert. Professionally, I work as a computer programmer in natural language processing and artificial intelligence. About 5 years ago, I decided to tackle a problem that had long puzzled me. With all of the very specific details that go into the formation of an animal or plant, it is just not possible for the protein encoding genes to encode all of this structure. There are two problems. First, 20,000 genes are not enough. Second, proteins are the machinery of the cell. It just does not make sense that the genome would encode the layout of the body in the machinery of the body. That would be like building your road map into the mechanism of the steering wheel. But the genome contains hundreds of thousands of transposons, jumping genes, and huge sequences of long non-coding DNA that is almost but not quite repeating, just like the image stored on a hard drive. So some morphogen proteins, like the Hox genes, would specify the gross structure in an analog fashion using morphogen gradients, but the fine structure is digitally encoded in the genome in the long non-coding DNA, which is manipulated by the transposons. This is similar to a computer hard drive. The proteins are the operating system of the body. They perform low-level functions, but the detailed structure comprises most of the other 99% of the genome. This detailed structure does two things. First, it specifies the structure and voids in a body part by commanding unneeded cells to self-destruct via apoptosis. The other cells are specialized by controlling the gene regulatory network. So an undifferentiated stem cell is put into place by morphogenesis, but as the cells are further specified as pluripotent and fully differentiated cells, their function is controlled through a tree structure of details that is turned on and off through methylation. This starts with the CPG regions, which can be found near the morphogenic genes such as the Hox genes. People think that the transposons are parasitical sequences that cause mutations, but their actual purpose in the genome is to perform a kind of origami on the DNA through to determine a three-dimensional fine structure of the body. This is done in tandem with the long non-coding DNA. This decoding is done in the chromatin. Euchromatin is loosely packed so that it can be unspooled to decode protein structure, but the heterochromatin decodes the structure encoding. It is tightly packed because all you need to do is determine the local context to figure out if a cell should go into apoptosis, or, if not, which part of the gene regulatory network should be switched on. This is specified in part by the histone codes. To find out more, read my article entitled Structure Encoding in DNA in the May 2020 issue of the Journal of Theoretical Biology.